Welcome back to the deep dive. Today, we're shortcutting your path to really understanding one of life's biggest mysteries. How does a single fertilized cell become, well, a whole complex animal?
Yeah, we're looking at the genetic program, the instructions that control animal development. It's incredibly precise.
And it's super relevant today, right? I mean,
Yeah.
think about all the buzz around stem cell therapy.
Absolutely. Parkinson's, diabetes, arthritis, the potential treatments all come back to understanding how cells specialize. How does one cell decide to become, say, a neuron versus a skin cell? That's development in action.
The stakes are definitely high. So our mission today is to synthesize the key ideas about the genetic control of development. We'll
Hmm.
be leaning heavily on the classic model system.
Drosophila melanogaster, the fruit fly. It's where so much of this was worked out. Development is really this intricate dance of genes turning on and off at just the right time.
Okay, let's dive in. Let's start with the fly itself. Why Drosophila?
Well, several reasons. They breed really fast, they're easy to handle in the lab, and you can manipulate their genetics relatively easily. Plus, crucially, they're basic developmental genetics.
Hmm.
It turns out to be remarkably similar to ours.
So how did researchers actually figure out the pathways? What was the strategy?
It was classic genetics, really. You start by looking for things that go wrong. Find mutations that disrupt development. Maybe a fly is missing a whole body section or grows legs where its antenna should be.
Wow.
Then you test if different mutations affect the same gene. That's allelism testing. You map where the genes are. And importantly, you do epistasis testing.
Which tells you the order, right? Which gene acts first in the pathway?
Exactly. It lets you build a kind of flowchart of genetic control. Who tells who what to do?
Okay, so let's look at the very beginning. The early Drosophila embryo is, well, it's pretty unusual.
It really is. After fertilization, the nucleus starts dividing like crazy. Rapid divisions. But here's the weird part. The cell membranes don't form immediately.
So you just have loads of nuclei floating around in one big shared cytoplasm.
Right. Precisely. That's called a syncytium. It's like one giant cell with many nuclei. They're all identical at this point.
Huh. Why do that?
It allows for really rapid communication and distribution of molecules from the mother before individual cells are walled off. After about 9 divisions, the nuclei move to the outer edge, forming the syncytial blastoderm,
Still sharing that cytoplasm.
still sharing. It's only around division cycle 13 that membranes finally grow down from the surface and enclose each nucleus, creating the cellular blastoderm.
And that's when you get distinct cells for the first time.
Right. This process separates the cells that will become the main body, the somatic tissues, from a small group at the back end, the Bol cells.
And the Cole cells are special.
Very. They're put aside early to eventually become the germ line, the sperm or eggs of the adult fly.
Okay, so that syncytial stage is like a developmental head start. What about later stages? You mentioned wings and legs earlier.
Right. So the fly larva hatches, eats, grows, then forms a pupa. Inside the pupa, metamorphosis happens. But the adult structures, like wings, eyes, legs, they don't just appear out of nowhere.
They come from those imaginal discs, right?
Exactly. Small groups of cells that were set aside, sequestered way back during the larval stage. They just sit tight, then expand and differentiate during metamorphosis.
Which brings up a key point. If the embryo hasn't even turned on its own genes yet in that early syncytial stage, and these discs are set aside early, how does the egg know front from back, top from bottom?
Ah, great question. That's all down to maternal control. The mother sets up the whole initial coordinate system before the egg is even fertilized.
Okay, so we're talking about maternal-effect genes. What's the key thing to understand about those?
The absolute key is that the offspring's appearance, its phenotype, is determined solely by the mother's genetic makeup for that gene. It doesn't matter what genes the father contributes, or even what genes the offspring itself inherits.
It's all about what the mother packed into the egg.
Exactly. She loads the egg with messenger RNAs and proteins during egg formation. The classic example is the dorsal gene, or DL. If a mother fly has 2 mutant copies of dorsal, all her offspring will fail to develop normally on their ventral side, their belly side.
Even if they get a working copy from their dad.
Even then, the damage is done because the mother couldn't provide the essential dorsal product in the egg cytoplasm. The instruction manual was faulty from the start.
Wow. Okay, so let's trace how these maternal factors set up the body axis. First, the dorsal ventral axis, top to bottom. You mentioned the dorsal gene product.
Right, so the dorsal protein is actually a transcription factor. And interestingly, it's initially present throughout the entire egg cytoplasm, uniformly distributed.
But it only acts ventrally, right? How does it know where the ventral side is?
It's guided by a signal on the outside. There's a signaling pathway involving proteins called toll and Spätzle. Think of the toll protein as a receptor embedded in the egg membrane. And Spätzle is the signal, the ligand. But Spätzle is only activated on the ventral side of the embryo by another protein, an enzyme called ester.
So only active Spätzle on the ventral side, like a little flag saying this way is down.
Exactly. When active Spätzle binds to the toll receptor ventrally, it triggers a signal inside the cell. And that signal's job is to tell the dorsal protein, okay, you can now enter the nucleus, but only here, on the ventral side.
So dorsal protein floods into the nuclei just on the bottom of the embryo. What does it do once it's in there?
It turns genes on and off. Where dorsal concentration is high in the nucleus ventrally, it switches on genes like twist and snail. These genes tell those cells you're going to become mesoderm, which later forms muscle and blood.
And where it's absent from the nucleus on the dorsal side?
There, it can't repress certain other genes like zircnolt and decapitoplegic. So those genes are expressed dorsally, telling those cells to become the epidermis, the skin. It sets up that fundamental top-bottom difference.
That's really elegant. Okay, what about the other main axis, anterior, posterior, head to tail?
This is set up by different maternal factors, primarily two key proteins acting as morphogens, Bicoid and Nanos.
Morphogens, meaning their concentration matters.
Precisely. A morphogen tells a cell what to become based on how much of the morphogen it sees. In the fly egg, the mother anchors the messenger RNA for Bicoid at the anterior, the future head end.
Okay.
And she anchors the Nanos mRNA at the posterior, the tail end.
So when these RNAs are translated into proteins, you get a high concentration of Bicoid protein at the front fading towards the back.
Right, an anterior to posterior gradient.
And a high concentration of Nanos protein at the back fading towards the front.
Exactly, a posterior to anterior gradient, opposing gradients.
So you have high Bicoid up front, high Nanos in back. How do they establish the pattern in between? Seems like they need to interact somehow.
They do, but indirectly, through regulating two other factors, hunchback and caudal. Their RNAs are initially distributed pretty much everywhere. Now, the Bicoid protein up front does two things. It acts as a transcription factor to boost the production of hunchback protein. But, critically, it also binds to caudal messenger RNA and prevents it from being translated into caudal protein in the anterior.
Ah, so Bicoid tells the front, make hunchback block caudal.
You got it. Now, down at the posterior end, the Nanos protein does the opposite job for hunchback. Nanos binds to hunchback messenger RNA and triggers its degradation.
So Nanos tells the back, get rid of hunchback RNA. It doesn't seem to affect caudal directly.
Not directly, no. But the result of these actions is clear. Because Bicoid blocks caudal translation up front, caudal protein only gets made in the posterior. And because Nanos degrades hunchback RNA in the back, hunchback protein is mostly restricted to the anterior.
So you end up with hunchback protein concentrated at the front and caudal protein concentrated at the back.
Exactly. And these two proteins, hunchback and caudal, are themselves transcription factors. Their opposing gradients provide the initial broad zones that tell the embryo where the head, thorax, and abdomen regions should form. It's the first major step in AP patterning, all set up by mom.
Incredible. So the mother lays down these broad Bicoid Nanos gradients, which then set up hunchback caudal zones. At what point does the embryo's own DNA start contributing?
Right about here. This is the transition to zygotic gene expression. Those maternal factors like dorsal, bicoid, hunchback, caudal, they act as switches to turn on the embryo's own genes in specific patterns. Dorsal activating twist in snail ventrally is a perfect example of a zygotic gene being switched on by a maternal factor.
And this kicks off the segmentation process, making the body segments.
Yes, and it happens in this beautiful cascade, a hierarchy of gene activity that progressively refines the body plan. It starts broad, then gets narrower and narrower.
Okay, what's the first level?
First up are the gap genes, like cripple and giant. They're switched on by the maternal gradients and the early zygotic factors like hunchback. They define large, multi-segment regions.
And mutations in gap genes cause...
Well, gaps.
Literally, a mutation might wipe out several adjacent segments, leaving a big hole in the larval body plan.
Okay, so gap genes define big zones. What's next?
Next are the pair rule genes, like fushi tarazu, which means too few segments in Japanese, and even skipped. These are regulated by the DAP genes. They get expressed in a really striking pattern of 7 stripes, perpendicular to the AP axis.
7 stripes. Why 7?
Because they define the boundaries of every other segment, or more accurately, parasegment. So they divide the embryo into 14 parasegments initially.
And a mutation in a pair rule gene.
Causes the deletion of every other parasegment. You get larvae that are like half the normal length with alternating segments missing.
Wild. Okay, gap genes, pair rule genes. What's the final refinement? The segment polarity genes, like engrailed and wingless. These are regulated by the pair rule genes, and they get expressed in stripes too, but now it's a 14-stripe pattern. Their job is to define the anterior versus posterior compartment within each individual segment.
So they give each segment its own internal front and back.
Exactly. And mutations here are really bizarre. Often, part of a segment is deleted, and it's replaced by a mirror image copy of the remaining part. It messes up the polarity within the segment.
Okay, so that cascade sets up the segments. Gap, pair rule, segment polarity. But a thorax segment is different from an abdomen segment. How do they get their unique identities?
Now we get to the homeotic genes, also called selector genes. These are the master regulators that tell a segment what it should become.
These are the ones responsible for those really dramatic mutations, right?
The most famous ones, yes. A homeotic mutation is one where one body part transforms into the likeness of another. The classic is bithorax. Mutations in the bithorax complex can cause the third thoracic segment, which normally had little balancers called halters, to develop like the second thoracic segment.
Which has wings, so you get a fly with 4 wings.
Essentially, yes, although the extra pair is usually rudimentary. Another famous one is antennapedia. Certain mutations cause legs to sprout from the head socket where the antenna should be.
Legs for antenna, that's amazing. So these genes are like master identity switches. Where are they located?
They're grouped together on the chromosome in 2 main clusters. The antennapedia complex, ANTC, which primarily controls identity in the head and anterior thorax, and the bithorax complex, BXC, which controls the posterior thorax and abdomen.
And what kind of proteins do they make?
They all encode transcription factors. And they share a characteristic DNA binding domain, a sequence of 60 amino acids called the homeodomain, which folds into a shape called a helix-turn helix. This domain lets them bind to specific DNA sequences and regulate batteries of downstream genes that execute the build-a-wing or build-a-leg program.
Okay, so they're high-level regulators. Is there anything special about how they're arranged in those clusters?
This is one of the most mind-blowing discoveries in developmental biology. The order of the genes along the chromosome in both ANTC and BXC perfectly matches the order of the body segments, from anterior to posterior, that those genes control.
Wait, seriously, the gene for the head part comes first on the DNA, then the gene for the next segment, then the next.
Exactly. It's called collinearity. The physical map of the genes on the chromosome mirrors the spatial map of their function along the embryo's body axis.
Why? Why would evolution do that?
That is a huge question. It suggests some very deep, ancient organizational principle, maybe related to how the genes are regulated during development, opening a chromatin in sequence or something. It's still an active area of research, but the discovery itself was just stunning.
Collinearity. That's wild. Okay, let's shift gears a bit. We've talked about the overall body plan. What about forming specific organs, like an eye?
Good transition. Organ development often relies on similar principles, including master regulatory genes. For the Drosophila eye, a key gene is called eyeless, or eye.
Eyeless? Does a mutation mean no eyes?
Pretty much, yes. The eyeless gene acts like a master switch. When it's turned on in the right cells, it activates a whole network, probably thousands of downstream genes, that are needed to construct a compound eye.
A single gene kicking off eye construction, that sounds important. Does it have counterparts in other animals?
It absolutely does. And this is where the evolutionary story gets really compelling. Researchers found the equivalent gene in mice, called PAX6, and they did this incredible experiment.
What did they do?
They took the mouse PAX6 gene and put it into a fruit fly. They engineered the fly so that the mouse gene would be turned on in, say, the cells destined to become a leg.
And what happened? Did the fly grow a mouse eye on its leg?
Not a mouse eye, but it grew an extra fly eye, a compound eye, structurally normal for a fly, just in the wrong place, like on its leg or antenna.
Wow. So the mouse gene could trigger the fly's eye-building program.
Exactly. It means the signal, the command build an eye, is so fundamentally conserved that the mouse version of the master switch works perfectly well to activate the fly's downstream eye-building genes. The common ancestor of flies and mammals, maybe 500 billion years ago, must have used this gene for eye development.
That's incredible evolutionary depth. Does this gene exist in humans?
Yes, we have a PAX6 homolog too, and mutations in human PAX6 cause a condition called aniridia, where the iris of the eye is partially or completely missing. It underscores that deep conservation.
Okay, so master genes are crucial. The eye also shows another key concept, right? Cell-to-cell communication.
Definitely. Specifically, induction. That's when one cell, which has already started differentiating, sends a signal to its neighbor, telling that neighbor what fate it should adopt. The development of individual facets, or ommatidia, in the fly eye is a perfect model for this.
How does that work?
Each ommatidium contains several cell types, including photoreceptor neurons labeled R1 through R8. They differentiate in a specific sequence. The last one to differentiate is the R7 photoreceptor.
And R7 needs a signal to become R7.
It does. It needs a signal from the cell right next to it, the R8 cell, which is already differentiated.
Okay, so R8 sends the signal.
Right. The R8 cell displays a protein on its surface called BOSS, which stands for Bride of Sevenless. That's the signal, the ligand.
And R7 has the receptor.
Yes, the R7 precursor cell has a receptor protein in its membrane called SEV for sevenless. This SEV receptor is a specific type called a tyrosine kinase.
So BOSS on R8 binds to SEV on R7.
And that binding activates the SEV receptor inside the R7 cell. It triggers a signaling cascade that ultimately tells the R7 cell's nucleus, okay, differentiate into an R7 photoreceptor.
What happens if you mutate either BOS or SEV?
If you have a loss of function mutation in either the gene for the signal BOS or the gene for the receptor, FREV, the R7 cell never gets the instruction. It fails to become a photoreceptor and instead differentiates into a different cell type, usually a non-neuronal cone cell. It shows how critical that specific interaction is for determining cell fate.
That precise communication is key. Now, how do these principles we've seen in flies apply to us, to vertebrates?
Well, as we saw with eyeless PAC6, the fundamental genes are often conserved. Researchers use the fly homeotic gene sequences, especially that conserved homeodomain, to search through vertebrate DNA databases.
And they found them.
Oh, yeah. They found vertebrate Hox genes, which are the direct homologs of the fly's ANTC and BXC genes.
The ones that control segment identity.
The very same. In mammals like mice and humans, there was likely an ancient duplication of the entire cluster, maybe even twice. So instead of 1 or 2 complexes, we typically have 4 clusters of Hox genes, usually labeled HOXA, HOXB, HOXC, and HOXD.
4 sets. Does that make studying them harder?
It adds complexity, for sure. But we can study their function using techniques developed mainly in mice. Things like generating insertion mutations, or more powerfully, knockout mutations.
Knockouts. That's where you specifically disable one particular gene.
Exactly. You target a gene, break it, and then see what happens to the mouse's development. For example, if you knock out the HOXA8 gene in a mouse,
What happens?
HOXA8 normally helps define thoracic vertebrae. Without it, you get a transformation. The first lumbar vertebra starts to look like a thoracic vertebra, and the mouse develops an extra pair of ribs. It's a segmental identity shift, just like the homeotic mutations in flies. It shows the principle holds.
So the basic toolkit is conserved. This all ties back to our starting point controlling cell fate for things like stem cell therapy.
Right. And here, it's important to distinguish between different types of stem cells. We have adult stem cells in many of our tissues, like in bone marrow or skin, whose job is to replenish specific cell types throughout life. They're generally multipotent, meaning they can make a few related cell types.
But the ones that get the most attention are embryonic stem cells, right?
Yes, embryonic stem, ES cells. These are typically derived from the inner cell mass of a very early embryo, the blastocyst stage. The key difference is that ES cells are pluripotent.
Meaning they can become
Pretty much any cell type in the body. Derivatives of all 3 primary germ layers. Ectoderm, like skin, neurons, mesoderm, muscle, blood, and endoderm, gut lining, liver. That huge potential is why they're so powerful for research and potentially therapy.
And this pluripotency is linked to the whole idea of cloning.
It is. We need to separate therapeutic cloning from reproductive cloning.
Okay, what's the difference?
Therapeutic cloning uses a technique called somatic cell nuclear transfer, taking the nucleus from a patient's body cell, say a skin cell, and putting it into an egg cell whose own nucleus has been removed. The goal is to get that egg to develop into an early embryo from which you can derive ES cells that are genetically identical to the patient. These could theoretically be used for transplants without immune rejection.
So the goal is cells, not a whole individual.
Exactly. Reproductive cloning, on the other hand, uses the same starting technique, but aims to implant that reconstructed embryo into a surrogate mother to develop into a complete live-born individual genetically identical to the nucleus donor, like Dolly the sheep.
But reproductive cloning, it often goes wrong, doesn't it? The clones often have health problems.
Very often, yes. They frequently suffer from developmental abnormalities, premature aging, obesity, cancer. It's technically very difficult and ethically fraught.
Why is it so hard to get it right if the DNA is identical?
It comes down mostly to epigenetics. Remember, that skin cell nucleus used for cloning came from a cell that was highly specialized. It had spent years turning certain genes off permanently, using chemical tags on the DNA, like methylation or modifications to the proteins DNA wraps around, the histones.
Ah, so the epigenetic memory of being the skin cell needs to be wiped clean.
Completely reset to the state of a fertilized egg, a totipotent state. And that process, reprogramming the epigenome, is incredibly complex and inefficient. The egg cytoplasm tries its best, but it often fails to erase all those specialization marks correctly. Those residual epigenetic errors are likely responsible for many of the developmental problems seen in clones.
So the DNA sequence isn't enough. The epigenetic state is crucial for proper development. That really highlights how complex control is. Now, for our last topic. There's a major exception to the rule that all our body cells have the same DNA sequence, right?
Yes, a spectacular exception. The cells of our immune system, specifically B cells and T cells.
What's the problem they solve?
The problem is diversity. Your immune system has to be ready to recognize and fight off millions, maybe billions, of different potential pathogens, viruses, bacteria, each with unique molecules on its surface. How could you possibly encode a specific receptor for every single one in your germline DNA? You'd need far more genes than we actually have.
Yeah, the numbers wouldn't work. So how do we generate all that diversity in antibodies and T-cell receptors?
Through a unique process that happens during the development of each individual B cell or T cell. It's called somatic recombination.
Somatic meaning in a body cell, not the germline, and recombination meaning rearranging the DNA.
Exactly. The genes that encode antibodies, for example, aren't present in the germline DNA as single, complete coding sequences. They exist as collections of gene segments.
Like building blocks.
Precisely. Let's take the kappa light chain of an antibody as an example. In the germline DNA, you have multiple different versions of the viral region segment. Let's call them loose segments, maybe 40 functional ones. Further downstream, you have several joining segments, G segments, maybe 5 of those. And then finally, a single constant region segment, C.
So lots of V options, several J options, one C option.
Right. Now, as a B cell is developing, specialized enzymes, including RAG1 and RG2 proteins, come in. They recognize specific DNA sequences called recombination signal sequences, RSS, that flank these V and J segments.
What do the RG proteins do?
They act like molecular scissors and tape. They randomly pick one of the 40 V segments and one of the 5 J segments. They then cut the DNA and precisely join that chosen V segment directly to that chosen J segment.
And the DNA in between, all the other V's and J's.
It gets looped out and permanently deleted from the chromosome in that specific B cell and all its descendants.
Whoa. So each B cell physically changes its own DNA to assemble a unique antibody gene.
That's exactly what happens. And think about the combinatorial possibilities. Just for the kappa light chain, you have 40 V choices times 5 J choices. That's 200 possible VJ combinations right there.
And that's just one part of the antibody. The heavy chain does something similar.
Yes, the heavy chain gene also rearranges using V, D diversity, and J segments, generating even more combinations. Plus, the joining process itself can be imprecise, adding extra variation. When you combine a randomly assembled heavy chain with a randomly assembled light chain, the total number of different antibody molecules a single person can potentially make is enormous, billions easily.
So the immune system literally cuts and pastes its own genes to create diversity after fertilization. That's a completely different strategy from the rest of development.
It's a brilliant evolutionary solution to the problem of pathogen diversity. It allows the immune system to generate a vast repertoire of receptors from a limited number of inherited gene segments.
What an amazing journey through developmental control. Okay, let's try to recap the big takeaways from this deep dive.
Sure. I think the first point is the hierarchy. Animal development starts with maternal control, where the mother loads the egg with factors like Bicoid and Nanos that set up the main axis via concentration gradients.
Right. Then those maternal factors activate the embryo's own zygotic genes, kicking off that cascade of segmentation, genes, gap, pair rule, segment-polarity that progressively divide up the body.
Then homeotic selector genes step in to give each segment its unique identity, and remarkably, these genes often show collinearity. Their order on the chromosome matches their order of function along the body.
We also saw the power of master regulatory genes like eyeless PAC6, whose conservation across vast evolutionary distances is just stunning. And how specific cell fates are often determined by cell-to-cell signaling or induction, like the R7 photoreceptor.
And we touched on vertebrate Hox genes, Stem cells, the difference between pluripotent ES cells and adult stem cells, and the epigenetic challenges involved in cloning.
And finally, that fascinating exception, the immune system's use of somatic recombination to physically rearrange DNA in developing B and T cells, generating the vast antibody and receptor diversity needed to fight infection.
Which leads me to maybe a final thought for you to ponder. We usually focus on the stability of the genetic code cast down through generations via the germline. But the immune system highlights the power of programmed genetic change happening within somatic cell lineages during an individual's lifetime.
Hmm.
So consider this. How does that ability for controlled somatic genetic alteration contribute to the organism's overall strategy for survival and adaptation? It's not just about inheriting a fixed blueprint, but also about having systems that can generate novelty and flexibility within a single lifespan. It's a different kind of developmental potential, isn't it?
That's a really profound point to end on. The genome isn't just static. In some contexts, it's dynamic even within our own bodies. Fantastic. Thank you for joining us for this deep dive into the genetic control of development.
And a warm thank you from the Last Minute Lecture team. We really appreciate you tuning in and dedicating your time to learning with us.